to understand chemotherapy classification and the mechanism of action, we should make a little bit deep diving to know more about mitosis and meiosis. As you can see here, this is the mitosis. The parent cell DNA replicates and give two daughter cells. And when it comes to meiosis, parent cell DNA replicates two daughter cells, and after that, the end result is 4 daughter cells. We should notice that most of the time when people refer to cell division, they mean mitosis, the process of making new body cells. Mitosis is a fundamental process for life. During mitosis, a cell duplicates all of its contents, including its chromosomes, and splits to form 2 identical daughter cells. Because this process is so critical, the steps of mitosis are carefully controlled by certain genes. When mitosis is not regulated correctly, health problems such as cancer can result. When it comes to meiosis, meiosis is the type of cell division that creates egg and sperm cells. Ensures that humans have the same number of chromosomes in each generation. It is a 2-step process that reduces the chromosome number by half, from 46 to 23, to form sperm and egg cells. When the sperm and egg cells unite at conception, each contributes 23 chromosomes, so the resulting embryo will have the usual 46 chromosomes. Review of cell cycle. This slide is quite important, and we should exert our utmost effort to understand every single point in this slide. The process of events that a cell goes through in order to divide. An understanding of the cell cycle is helpful to understand the chemotherapy. As you can see here, cell cycle starting from G0 resting phase, cells that are not active. And here, as you can see, cell cycle non-specific drugs are working in this stage. Alkylating agents, anti-tumor antibiotics, platinum agents. And when it comes to G1, in G1, the cell is preparing for cell division by upregulating synthesis of proteins, enzymes, macromolecules, and organelles. S synthesis phase. In this phase, the cell replicates each chromosome. As you can see here, the S phase specific drugs such as antimetabolites and topoisomerase inhibitors. G2, or second growth phase, involves growth prior to mitosis. In the G2, bleomycin is G2 phase specific drug. For the M mitosis phase, as you can see here, finally M phase is where the cell divides through the process of mitosis, and vinca alkaloids and taxanes are M phase specific drugs. The cell cycle, as you can see here, starts on the G0 resting state, cell is not dividing. After that, G1, synthesis of enzymes needed for DNA synthesis. After that, S phase, DNA is replicated, and in the G2, synthesis of cellular components required for mitosis. And finally, M, mitotic phase, cell divides. Then back to G0. Here, cell cycle specific drugs examined: antimetabolites, bleomycin, vinca alkaloids, and etoposide. They are effective for the high growth fraction malignancies such as hematologic cancers. And for the cell cycle non-specific drugs such as alkylating agents, antibiotics, cisplatin, and nitrosourea, effective for both low growth fraction malignancies such as solid tumors as well as high growth fraction malignancies.